In chapter 1 of our investigation of microbiology in our Roberts Wesleyan University microbiology course, we looked at what types of organisms we would be considering in microbiology, things that are too small to observe with the naked eye. And we also looked at a brief history of microbiology. So today we're going to take up different ways of making those microscopic objects visible, different types of microscopy, and kind of just very briefly how they work, the principles they're based on. So this graph is a little bit tiny to visualize, but you can use it as a reference later as we're talking about different types of microscopy. It shows you on the x-axis a log scale of sizes of objects or distances, and then plotted above that is the range that different types of microscopes can resolve or see. And so you can see over here the unaided eye can see down to around 100 micrometers under ideal conditions. Light microscope can allow us to resolve or visualize objects much closer together down to a resolution of around 100 nanometers. And then the most, in this particular graph, the transmission electron microscope can see down to around 10 picometers. And now we're getting into some very small territory indeed. We'll say more about what those different units mean in the next slide. So oftentimes when we're talking about just measurements in everyday life, we talk about meters, centimeters, millimeters. Then we can go down in orders of magnitude 3 at a time and define different units of measure. So if a millimeter is 1,000th of a meter, a micrometer is 1,000th of a millimeter, so 3 decimal places down. A nanometer is 1/1000th of a micrometer, and a picometer is 1/1000th of a nanometer. So we're getting down into some very, very small stuff by that point. So just on these last two little lines are to help you mathematically express these things. If a nanometer is 1,000th of a micrometer, that means there are 1,000 nanometers in every micrometer. Or how much of a micrometer is 1 nanometer? 0.001. 1/1000th of a micrometer is a nanometer. We already took a look at this brass microscope that Van Leeuwenhoek had made. There's a little glass lens right in here, and the sample on the end of this pin with adjustable height and adjustable distance from the lens with these different thumb screws. And it's called a simple microscope because it only has one lens. Light enters from behind this object, or actually light enters from our point of view looking at this slide, and then the viewer would be on the other side of the microscope capturing that light and analyzing the subject. Whereas typically the microscopes we use have two different lenses that together produce a magnification that is the product of their two individual magnifications. So the objective lens here in a compound microscope magnifies the light traveling up through our specimen on the stage. We have a light source down here, a condenser lens to even out the brightness of the light source and so forth. Let that nice uniform beam of light hit our specimen and then travel from there into the objective lens, which magnifies it. And then that magnified light is sent through an ocular lens, which further magnifies it, and that's what we see with our eyes. So the total magnification is ocular times objective magnification. We'll look at a few different types of microscopy. We'll look at all these listed here are light microscopes. They use light, which as we'll see later limits the resolution or how small of an object can be resolved by these types of microscopes. So first let's look at a compound light microscope. We've already looked in that cross section at one. We have a light source down here at the base, a condenser lens, send a nice evenly illuminated beam of light up through the specimen on the stage, and then into the objective lens. You can see this microscope has multiple different objective lenses with different magnifications that can be applied by rotating this turret. Then the light travels using a prism to change its direction and send it through an objective lens to the eye. So resolution is the ability of a microscope or a lens to distinguish between 2 points. The smallest distance between 2 points that can be distinguished by the lens is called its resolution. So in other words, the closer the points are that we can tell apart, the smaller the features of our sample we can recognize. It turns out that the wavelength of light is what limits the resolution of a microscope or a lens. The resolution can't be a smaller distance than the wavelength of the light. As those photons of light travel through space, they have a wave characteristic, and if the wave characteristic limits the resolution, visible light is what limits the light microscope. There are other things that improve resolution, though, besides just the wavelength of light being a limiting thing. Contrast. If there's no contrast, we can't tell things apart from each other. Most microorganisms and tissues in the body, for that matter, in a light beam, have very little contrast. In other words, they're essentially transparent. So adding contrast will increase the resolution of our equipment or our microscope. So what is refractive index? When light travels from one medium into another, from liquid into air, from air into liquid, from air into glass, glass into air, every time the light crosses a boundary between different media, it changes direction, because as it changes speed, that wave characteristic bends the ray of light. That's called refraction. Refractive index is a measure of how fast the light essentially can travel in that medium compared to in a vacuum, which is when light rays travel the fastest. Anytime you put some stuff in its way, kind of slows it down. And so if light crosses a boundary between two substances of an identical index of refraction, it won't bend at all. But if there's a very big difference in index of refraction, then the light will bend a lot. We'll have a change of direction. And for the highest magnification objective lenses for light microscopy, the highest magnification, 100 times magnifying objective, will require a drop of oil placed between the sample and the objective lens, so that the light rays that are refracting as they pass from the sample into the air won't be lost by the objective lens. The oil keeps the index of refraction between glass to oil back to glass again, like say a glass slide, keeps it very similar, and the light doesn't refract very much. We can catch those rays of light into the objective. Here's a little diagram, right? So we have a high magnification objective here. Light passing through the condenser lens is coming to our sample, and it will refract in this particular objective. Has a very narrow opening here, can't capture very much light unless we can prevent it from bending and escaping. We'll keep that immersion oil keeps the light ray coming right straight up into that objective lens. So the 100x objective in your light microscope in the microbiology lab will require a drop of immersion oil. Without it, you just see a fuzzy image. A lot of the light is refracted and lost. Brightfield illumination is a specialized light microscope method that makes objects appear dark against a very bright background. So light reflected off the specimen doesn't enter the objective, so it creates a dark area where the sample was, and then we'll see it against a bright background. So we're increasing contrast, which helps increase resolution. Specimens have to be fixed and stained. We haven't said much about fixing and staining. I don't think we did yet. If we don't come upon a slide about fixing and staining soon, I'll run through that with you. So here's a picture of a Brightfield view of a paramecium or some related microorganism. Darkfield illumination does just the opposite. We see a bright sample on a dark background. It's just a different way of arranging the way the light enters the sample and is collected. And in this case, we don't need to stain the sample. We can see this particular microorganism against this dark background just based on the way the light is collected. So light that is reflected by features of our sample are going to enter into the objective lens. Any light that came from the condenser that did not interact with the sample will be lost, hence the dark background. Again, so only light that's been impacted or changed in some way, reflected by features of the sample, will be collected, and that's why we see this bright subject in a dark background. Phase contrast is a very important one. Light field, bright field, and dark field we won't talk about a whole bunch, but phase contrast we'll use quite a lot in most biology applications. And in this particular situation, there's two light sources, and they're going to travel through the sample or not through the sample, and the light rays that travel through the sample will be impacted by the sample, refracted, and then when we recombine the two light paths, they will destructively interfere or constructively interfere to produce dark and light regions, and it'll create some contrast that way. And this is a very ingenious thing because we don't need to fix or stain our sample in order to be able to see it. So live specimens can be viewed harmless, unharmed in phase contrast illumination. And so here's a picture of a phase contrast image of, again, a microorganism. In this case, it wasn't stained at all. It was simply placed on the slide and introduced into this, let the light travel through a phase contrast objective in this light microscope. So we have light rays that have been refracted or scattered by the sample, and light rays that have not. We combine them after they pass through the sample, and they will interfere with each other in such a way that we get this pattern of features of the sample. Very cool. Differential interference contrast microscopy is just or Nomarski, it's also called, is just another way of applying a similar principle to phase contrast. It's again something that can allow unstained samples to be visualized with quite a lot of contrast and resolution. So it produces like a 3D image by using multiple light sources. It's a very beautiful thing. It's a little more involved, a little more high-tech than phase contrast, so it's less likely you'll encounter a microscope like this, although we do have one in our department. Fluorescence microscopy. This method improves the resolution over a regular light microscope by using ultraviolet light, which has shorter wavelength than visible light. So we're going to take ultraviolet light and shine it on the sample, and certain substances can absorb ultraviolet light, and it excites electrons in the chemicals so that when they relax back down to their base state of energy, they release light. So they're going to absorb some UV light, and they'll actually emit visible light. But because the UV light has a smaller wavelength than visible light, we'll get a higher resolution view of our sample. You actually observe the fluorescence coming out at a 90-degree angle to the source of illumination. Kind of a cool thing. So here's a little view of a sample. Oop, it disappeared again. I don't know what happens when I do that. I guess it doesn't really matter. So here are some spirochete bacteria that have been stained with a fluorescent dye that can absorb that UV light and emit a visible light, in this case a green wavelength of light. And so we see a beautiful high resolution image of those guys. Confocal microscopy is a type of fluorescence microscopy in which we're going to stain our sample with a fluorescent dye or fluorescent dyes, but instead of just shining the ultraviolet light at the sample, we're going to use a laser of the appropriate wavelength to excite the electrons and scan back and forth at a precise depth through the sample, and then change the depth a nano or a micron or so, and just keep on scanning at different depths through the sample and collect the data at each time through each pass, and create a 3D image of the illuminated sample. So it's a very high resolution and very, very powerful method because we can find the location of some feature within our sample in 3 dimensions. There's a picture of a confocal scanning microscope image of of the same microbe we've been looking at in several different slides. Two-photon microscopy is an interesting, more modern invention, even more modern. These are all relatively modern inventions, but in this case, um, um, long wavelength light is used to excite the electrons in some particular types of dyes, and then they will actually emit a shorter wavelength of light when they decay back down to their surface energy. And the beauty of this method is the longer wavelength illumination light is not scattered very much as it passes through the specimen, and so we can observe features much deeper into a specimen than most microscopic methods. Methods we've been talking about look down at the micrometer level depth. This can show us well-resolved features down to a millimeter of depth in a tissue sample, for example. So it's a really cool method. And there's again a picture of a two-photon image right there, where we're seeing quite a bit of the depth of all the features in that paramecium or whatever it is. I'm not going to spend time on some modern methods that use a microscopic tipped probe to resolve things down to the atomic level. They're not something that you're going to see in a microbiology lab. These are theoretical or research kind of grade instruments that are used for very cool stuff for observing the shape and position of things at the atomic or molecular level. Very, very cool stuff, but we'll just pass right by. 1 micrometer resolution for the scanning acoustic microscopy, which uses sound waves and reflection of sound waves to develop an image. Electron microscopy, there's a bunch of different, there's scanning tunneling microscopy. I can't remember if it's in this PowerPoint or not, but it's another one that can look at very, very small features indeed. Electron microscopy is fairly commonly used, but again, not on an everyday basis in a microbiology lab. In this case, the illumination source, instead of being visible light or ultraviolet light, is electrons. And electrons are charged particles that amazingly have wave characteristics, much like a photon. And so as they travel through space and they can be focused by electromagnetic lenses. They can't use glass lenses. They're much too high of energy. But we can use electromagnetic lenses to focus electrons after they pass through a sample and produce a very, very high resolution image because electrons have a very short wavelength. So here's a little schematic diagram of this tower-like electron microscope. We have an electron beam and we have some electromagnets, coils of wire wrapped around the place where the beam of electrons is going to pass down through. Electrons will pass through the specimen at some point in their travel, be focused some more, and they will strike a phosphor, a plate full of material that will be illuminated when the electrons strike them. And we can get an image that's reminiscent of our specimen on the stage there. The thing about electron microscopy is, first of all, in order to shine the electrons through, we need to evacuate the chamber of all air molecules. So we have to have a very, very strong vacuum inside here. So that right there makes it a rather esoteric thing, meaning kind of a rare thing to be able to do, expensive and complex. Another thing is that the electron beams have so much energy that if we just put our typical sample that we'd use in a light microscope on the stage, it would be burnt to a crisp in no time by the electron beam and we wouldn't see a thing. What we'll do instead is we will use heavy metal salts. Heavy metals are just great big atoms that can scatter the electron beam and allow us to see things. Where there's a heavy metal atom, we'll have a dark spot, and where there isn't, we will have a light area and so forth. So we can spray the sample essentially with a source of heavy metal salts or atoms and ions, and then let that dry, and then shoot the electron beam in there, and we'll get an image. That's for transmission electron microscopy. The electrons are passing right through the specimen, and here's a beautiful picture of a false color image of again a microorganism. Look at the level of detail in that thing. The resolution is just incredible. 10 micrometers, and we're talking about probably a 1 micrometer resolution of details that we can see in this picture. Quite astonishing. Here's another picture of just a non-enhanced micro resolution image of a mitochondrion, and you can see all the layers, the inner and outer mitochondrial membrane, all the folds of membrane in there. Just remarkable. Pretty cool stuff. You can see endoplasmic reticulum with all the ribosomes against it and so forth. Very beautiful, a little bit esoteric. A scanning electron microscope uses a beam of electrons that instead of shot through the sample is scanned back and forth across the sample, and as the electrons are reflected off of the heavy metals that are placed on the sample, or some dyes that are placed on the sample, they're captured by some sort of a collector that allows an image to be produced, and it produces a beautiful three-dimensional surface image of the object. I'm sure you've seen plenty of SEM images. If not, go on Google SEM. Just no end of delightful microscopic pictures of high resolution that can be found. Oh yeah, here's some very, very high resolution things that again are not really anything more than a curiosity for us in microbiology. A scanning probe microscope uses a metal probe with a tip that comes down to a single atom at the tip and can resolve down to 1/100th of an angstrom. An angstrom is 10 to the minus 10th meters, and we can see down to 1/100th of that. So we're looking at a protein here. All of the individual amino acid subunits can be resolved in this picture, which is truly astonishing. And here again we see another type of atomic force microscope, a type of scanning probe microscope that kind of can get a real 3D image. We can kind of poke way down into the spaces between these structures and see the shape of things. Okay, so that pretty much wraps up our discussion of different types of microscopy. Let's talk more generally about how microbiologists will actually conduct their staining and their microscope work. Staining, again, most structures that we're interested in looking at have no contrast of their own, and we need to add stains to make them visible in the microscope. Again, typically a light microscope or a fluorescence microscope. A smear. We'll take a sample that contains microorganisms that we want to identify or review and make them fairly dilute, and then put an ultra-thin film of this material on a glass microscope slide and let it dry. If you put too thick of a layer, or too viscous or thick of a layer on there, when you try to observe it in the microscope, you won't be able to resolve one individual bacterium and anything about it, or any protozoa or whatever it might be. They'll be piled up miles deep, and you won't be able to individually identify any features of one unit. So you want a very, very thin film smear of an appropriate dilution. Let it dry, and then you need to fix it, attach it to the surface of the slide. So first let it dry, then heat it by passing it slowly, about this speed, microscope slide through the flame of a Bunsen burner or a Fisher burner. And what that'll do is evacuate the last bit of water that's trapped between the microbe and the glass, and to make it stick right onto the glass. And now you can do lots of staining and washing steps without all the microorganisms falling off. So first you make a nice smear, and then you fix it, and then we're going to stain it with something to make it have some contrast. Basic dyes are positively charged colored chemicals. And most dyes that we use in microbiology are basic dyes because they stick to lots of negatively charged proteins and structures within our microbes. So that's the most common thing we're going to use, basic dyes. We have a simple basic dye, which is a single chemical. Acidic dyes have a negative charge, and they typically will not stick to our microorganism. They'll in fact stick to the background. So sometimes we can use an acidic dye to stain the background. This is called negative staining. Most of our color dyes that we use to actually identify or image an actual microorganism are basic dyes. Chromophore is another word for a chemical that has some sort of a color that can be increase the contrast of our substance once it sticks. All right, so let's look at simple stains are just single dyes that can be added to a sample, and then they'll stick to the sample. We can give a brief rinse, and that'll create some contrast between the dye stuck to our microorganism and the background, which will be lighter, and now we can see it in the light microscope. A mordant is a stain that will enlarge something, it'll coat or stick to something like the flagellum of a bacterium, which you can't see in the light microscope, make it thick, and then it can be visualized. So that's an idea of a mordant. Make something enlarge it so you can see it. Also, we'll see that a mordant is a word used to describe a capturing step in one of the staining techniques we'll see. Differential stains means that a stain is not just going to make microorganisms visible, it's going to distinguish between specific types of microorganisms. Maybe one particular type will stain and all the rest will not. Gram stain and acid-fast stain. Let's take a look at those two examples of differential stains. Gram positive and gram negative. Most microorganisms, most bacteria I should say, that's specifically bacteria, can be classified as either gram positive or gram negative based on the structure of their cell wall. Gram positive bacteria have a very thick peptidoglycan cell wall. Gram negative bacteria have a very thin peptidoglycan cell wall and an outer membrane. And when we use the stain, we're going to trap some dye in that thick peptidoglycan cell wall of the gram positive bacteria, and it'll be washed out of the gram negative bacteria. So now we have a differential stain to tell gram positive from gram negative bacteria. Here's what we're going to do. We're going to take a mixture of bacteria, or an unknown bacteria, stain them with crystal violet, a basic dye. It's going to go into the cells, and at this point, gram positive bacteria will look purple and gram negative bacteria will look purple. We're going to add a mordant, we're going to add iodine, and it's going to combine with the crystal violet and make large dye particles within the bacteria. Not going to change anything about the staining. And then we're going to do a decolorization step. Alcohol mixed with organic solvent acetone will wash the stain right out of a gram negative bacterium because it has that thin peptidoglycan cell wall. The stain can be washed right out. The stain particles with the mordant added can't fit through the tight mesh of the gram positive cell wall, which in fact will actually contract also during the decolorizing step and make the passageways in between the mesh of peptidoglycan even tighter, and the dye will be retained. So now those cells remain purple, whereas the gram negative cells are colorless. And we use what's called a counter stain, because now the gram negative cells are invisible. We wouldn't know if they're there or not. We'll use safranin, which is just a red basic dye, to make the gram negative cells stand out against their background, and we'll still be able to see the purple gram positive cells. And let's go right to a slide here, and we can see some these purple cells are gram positive bacteria, and the pink cells are gram negative bacteria. So this beautiful differential stain has been done to tell the difference. Cells that are bacterial cells that are elongated like this are called cocci, and these little spherical bacteria are called staphylococci. Boy, I had a little mental block there for a second. I'll say that again. I completely mixed up that slide. Cocci are spherical bacteria, and rod-shaped bacteria are these elongated ones. They're called bacilli. And sorry for that little absence of mind for a moment. Acid-fast staining is another type of differential staining in which a particular genus of bacteria, two genera here, Microbacterium and Nocardia, have a waxy compound in their cell wall, which makes them very, very hard to stain in the first place, but allows them to retain a stain under certain conditions that will not be retained by any other bacteria. So we have a differential stain for Mycobacterium, bacteria that cause tuberculosis and tetanus, for example, can be identified this way. So what we're going to do is use a primary stain, carbylfuchsin, which is red, and it's going to stain all the cells, as we saw before. We're actually going to heat and pound that red stain right into that waxy cell wall of our Mycobacterium. And then we're going to use a decolorizing agent that's very aggressive, acid alcohol, and it's going to remove the stain out of the non-acid fast cells. Any other genre than those two that we saw are going to become colorless, and our Mycobacterium will remain red. And then we'll counter stain with methylene blue, and here's our result. We can see Mycobacterium bovis is shown in these blue bacilli here, and the cocci are Staphylococcus, most likely staph bacteria that are gram positive, but they are not able to retain the carbylfuchsin. I shouldn't have mentioned gram positive here. I'm confusing you. All right, some other special stains: capsule stains, endospore stains, and flagella stains. A capsule stain is a negative staining method, typically used in combination with a simple stain to look for the capsule, a thick polysaccharide covering around the surface of a bacterium. If we just stained the bacteria with a simple stain, we would just see the bacillus or the coccus, and we wouldn't know if it has a capsule or not. And if we add a negative stain that stains the background, you can see, and here in this picture you'll see some bacilli here that are stained with a simple stain, which makes them turn pink. But then there's a background stain, and the white area is this thick polysaccharide capsule on the outside of the cells. Some species of bacteria that have a gene that allows them to produce a capsule are disease-causing, and when they don't have the capsule, they're not. So it's important to be able to recognize the presence of this capsule here. Sometimes it allows us to diagnose which of several possible bacteria is causing a disease, a particular disease like pneumonia. Endospores. What are endospores? Endospores are almost like little seeds that are produced by a couple of genera of bacteria, Clostridium and Bacillus. And those bacteria, if they're deprived or exhausted of nutrients in their environment, will eschew most of the content of the bacterial cell and shrivel it right down and produce a hard capsule around the remaining entity, including the genome, chromosome, and the thing will go into a dormant state. That's called an endospore. It's enormously tough, very hard to kill or destroy. And then when that endospore is placed in an environment where there's rich nutrients and water available, again, it will germinate and bacteria will grow once again. They'll be what's called vegetative or growing bacteria. So endospores are a big problem because they're hard to kill. Anyway, we can view endospores by using malachite green as a primary stain and heat, and that'll drive its way into that hard capsule of the endospore. Then decolorize with water, and that primary stain will not be retained by bacteria typically, and then counterstain again so we can see what bacteria are present. And here you can see some bacillus bacteria, and there's the endospores are shown in malachite green. And those are the little seed-like tough encapsulated dormant species that will survive some tremendously harsh conditions until it's time to germinate once again. And finally, flagella staining. Flagella are not visible in the light microscope. They're too thin, but if you add a mordant, a stain that makes them much thicker, then we can see them in this illustration. We see some. These are flagella. These are some structures on the bacteria that allow them to have motility. They can drive themselves through the water like a motorboat using rotation of these flagella. And now we've made them visible by using a mordant, or a Flagella stain, I should say. All right, so that's our story for viewing microorganisms in the microscope. I hope you enjoyed that chapter. It is pretty mind-bending to think of how small things we can see in different types of microscopes. But again, remember, in microbiology, on a routine basis, we're going to use compound light microscopes, and we're going to use fluorescence microscopes, dark field, bright field, phase contrast, and that's pretty much the extent of it. So fluorescence and bright, dark, and phase contrast microscopes.